Chapter 10, Section 3, Regulating the Cell Cycles. In multicellular organisms, it's very important that the cells do not grow out of control. And so in this section, we will discuss how they regulate growth. The cell cycle is controlled by regulatory proteins, both inside and outside the cell. The controls on cell growth and division can be turned on and off. For example, when an injury such as a broken bone occurs, cells are stimulated to divide rapidly and start the healing process. The rate of cell division slows when the healing process nears completion. Cyclins, which control cell growth and division, are a family of proteins that regulate the timing of the cell cycle in eukaryotic cells. In the graph below, you will see how the cyclin levels change throughout a cell cycle in a fertilized clam egg. In the graph, you should see that the concentration of cyclin is highest during the meiotic or the mitosis stages and low during the interphase stages, building to high. In this diagram, we look at how the cyclins change through the cell cycle. Remember that we start at the G1 or growth 1 phase. As the growth 1 phase progresses, as the cell reaches a certain size, then cyclin E is secreted, which causes the cell to go into the S or the division phase for the genetic material, the DNA. The DNA gets copied during the S part of the cycle. Once the DNA is copied, then cyclin A is released, which sends the cell into the G2 stage, which is the preparation for division. Once the cell is ready to divide, cyclin B is secreted, which starts the mitotic changes and it leads through to cytokinesis. At the end of cytokinesis, cyclin D is emitted, which causes the cell to go into the G1 stage, and it continues. The regulatory proteins come from two sources, internal and external. Internal regulators are proteins that respond to events inside the cell. They allow the cell cycle to proceed only once certain processes have happened inside the cell. So in other words, until the cell is ready to grow, it will not start duplicating its DNA until it's ready. External regulators are proteins that respond to events outside the cells. They direct cells to speed up or slow down in the cell cycle. An example of this would be, again, the broken bone. When the body detects the damage to the bone, then proteins are released that causes the cells around the damaged area to speed up their cell division or cell cycle, and they grow the number of cells grows very quickly. One type of external regulator is a growth factor, and they stimulate the growth and division of cells. They are important during embryonic development and in wound healing. In multicellular organisms, the number of cells that we have cannot grow past a certain point. And to prevent this from happening, each cell in our body has a program inside the genetic material that once a certain criteria is met, that the cells start to die off. This process is called apoptosis, and it's a programmed cell death. Apoptosis plays a role in development by shaping the structure of tissues and organs in plants and animals. For example, the foot of a mouse is shaped the way it is partly because the toes undergo apoptosis during tissue development. Sometimes our cells lose the ability to detect when things are getting out of hand. When this happens, we start to see things called cancers. Cancer cells do not respond to the signals that regulate the growth of most cells. As a result, cells divide uncontrollably. Cancer is a disorder in which body cells lose the ability to control cell growth. Cancer cells divide uncontrollably to form a mass of cells called a tumor. Typically, this happens when genetic information inside the cell is damaged in some manner. Sometimes we have a benign tumor or a non-cancerous tumor. It does not spread to surrounding tissue It does not harm surrounding tissue. A lot of times it's just extra cells that aren't needed. The different type is called a malignant tumor, which is cancerous. It invades and destroys the surrounding healthy tissue and can spread to other parts of the body. The spread of cancer cells is called metastasis. Cancer cells absorb nutrients needed by other cells, block nerve connections, and prevent organs from functioning. During normal regulation, the cells grow, and once they reach a certain criteria, they stop reproducing extras and just maintain. In a tumor, we provide extras. The benign tumor has a tendency just to expand, and typically with benign tumors, they're easily removed and don't need any more treatments. They typically don't come back. In the malignant tumors, they are where the cells are completely malfunctioning. They should have self-destructed through apoptosis, but did not. And so we have a damage to both the regulatory system in the cell and to the functionality of the cell and the ability to destroy itself when it doesn't work. Malignant tumors can go beyond the original site of the tumor and spread to other parts of the body. They interfere with the normal process of the organ that the cells make up. There are a variety of causes for cancer cells. We are in no way, shape, or form talking about every type of them. There are very, very many. The vast majority of them come from some type of a cause. Cancers are caused by defects in the genes that regulate cell growth and division. Some sources of genetic defects are smoking, tobacco, radiation exposure, defective genes, and viral infections. A damaged or defective p53 gene is common in cancer cells. It causes cells to lose the information needed to respond to growth signals. The p53 represents a particular gene and a location on that gene. When we treat cancers, we have three main processes we use. Sometimes they're used individually, sometimes in pairs, or sometimes all three of them are used in conjunction. Some localized tumors can be removed through surgery. Many tumors that are hard to get to and hard to work on through surgery, they are treated with radiation. The radiation damages the DNA inside the cell so it can no longer function at all, and the cell dies. There is a problem, though, in that radiation also causes damage to healthy cells. So it's taken very seriously when they do this. Chemotherapy is the third process we use, and that's where compounds are put into the body that kill the cell, and preferably the cancer cell, but has effects on other cells as well. They can also slow the growth of cancer cells.